Hi everyone, normal cells have tightly regulated growth and replication. Proto-oncogenes help the cell to grow and initiate the cell cycle. They include growth factor receptors, transcription factors, intracellular signal transducers. Tumor suppressor genes include p53 and retinoblastoma and help stop damaged, diseased, and mutated cells from replication. In its hypophosphorylated form, Rb binds E2F to inhibit the progression from G1 growth phase to S phase of the cell cycle. Tumor suppressor genes initiate cell apoptosis. Cancer cells proliferate uncontrollably, lacking anchorage dependence, contact inhibition, and growth factor dependence for proliferation. Genetic alterations can result in overactive oncogenes that promote cell proliferation or inhibition of tumor suppressor genes. Genetic alterations can be caused by chemical carcinogens, ionizing radiation, and microbes. Over 20% of cancers are caused by viruses. HTLV-1A human T-cell leukemia virus is a positively single-stranded RNA retrovirus which causes adult T-cell leukemia in a small percentage of patients 20 to 40 years after primary infection. Commonly found in Japan, Caribbean, Central Africa, and indigenous Australian communities, HTLV-1A can be spread by contaminated needles, sex, and breastfeeding. HTLV-1A binds the GLUT1 receptor of CD4 plus and CD8 plus T cells for entry. Subsequently, it induces a non-lytic response once integrated into the host genome and uses many transacting elevating elements such as Tax, HBZ mRNA, REX, and HBZ proteins. Upon glucose metabolism, hypoxia, stress, and cell activation, Tax protein is expressed. Tax activates cellular oncogenes, IL-2 and IL-2R genes, and upregulates HBZ mRNA, which together increase infected T cells to proliferate. Tax protein also promotes virion production, so more T cells are infected. During late-stage leukemia, HBZ protein is translated from HBZ mRNA. HBZ protein inhibits Tax mRNA to avoid the cytotoxic response. HTV-1A protein REX also plays a role in enhancing the efficiency of nuclear export of proteins from the cell. Hepatitis C is a positively single-stranded RNA flavivirus that causes liver inflammation and damage. Spread by contaminated needles, breastfeeding, and sex, it is common in Southeast Asia and North Africa. HCV replication can activate cellular protein kinase CPKR. CPKR inhibits p53 tumor suppressor gene. Hence, infected hepatocytes do not undergo apoptosis and continue to proliferate uncontrollably. Hepatitis B is a double-stranded hepatinovirus that causes hepatocellular carcinoma in 2 to 10% of infected persons. HBV is spread by contaminated needles, breastfeeding, and sex. HBV is common in Southeast Asia, North and Central Africa. HBV has an X gene which uses insertional mutagenesis to activate cellular oncogenes. HBV increases the accumulation of chromosomal mutations that may be oncogenic. Polyomavirus is a double-stranded DNA papavirus that is responsible for Merkel cell carcinoma. Merkel cells are oval skin dermis mechanoreceptors that play an important role in touch. After monoclonal integration of polyomavirus into Merkel cells, there is the expression of large tumor antigen and small tumor antigen to bind and render retinoblastoma factor and phosphatase 2A defective respectively. The loss of tumor suppressor gene results in overproliferation of Merkel cells with a characteristic flower appearance. EBV. Epstein-Barr virus is a double-stranded herpes virus that can cause nasopharyngeal cancers. EBV promotes translocation of chromosomes 8 and 14, combining the immunoglobulin heavy chain enhancer on chromosome 14 with the cellular oncogene c-MYC. Overactivation of c-MYC stimulates cells to enter the replication cycle. HPV, human papillomavirus, causes cutaneous benign skin warts or mucosal lesions including malignant cervical cancer caused by HPV-16 and HPV-18. In benign forms, HPV exists as circular DNA or episome. In malignant cases, HPV integrates into the host cell genome and may lose the E2 gene. The E2 gene usually inhibits E6 and E7 genes. Without E2, E6 and E7 are activated. E6 bind p53, promoting p53 degradation. E7 binds the RB pocket domains, which are essential to RB function. The inhibition of tumor suppressor genes means uncontrolled cell proliferation. These are the 6 main viruses that cause over 20% of cancer infections. That infects cells and account for over 20% of cancers. This is a summary table. Thank you everybody for listening.